Welcome to my mini lecture, and thank you for inviting me to explain to you my signal dissection method SDCM today. It is useful, for example, to reveal overlapping nonlinear interactions in large high-dimensional datasets like transcriptome measurements. My name is Michael Grau, and I'm currently researching in the aggressive lymphomas lab of Professor Dr. Med Georg Lenz at the University Hospital in Münster. My background is not medicine, though. I did my PhD in the Complex Systems and Biophysics work group of Professor Dr. Peter Lenz at the University in Marburg. In the first part of my mini lecture, I will now explain the problem we are trying to solve with my method. You see a microscopy image of cancer cells stained in blue with some invading blood vessels in red. And as you might know, cancer cells do not show natural cell death and proliferate uncontrolled in the body. If that's untreated, the patient dies. So we want to understand them on a cellular level and ideally stop them. For that, we measure them on a molecular level. Here you see a typical transcriptome data set. Every row is one gene or transcript, and every of the about 500 columns is one sample vector in that 55,000 dimensional transcriptome space. You have to zoom in quite a bit before you can see individual measurement values. Black means median gene expression, red above and blue below median gene expression intensity. To give you a familiar number, it's about 27 megapixels in size, but in contrast to your usual photography, it doesn't have any useful local neighborhood information, like large green forests, for example, and it shows a lot of random and systematic noise. More importantly, every tumor sample measures an ensemble of many different cells, and per cell we have many concurrent gene regulation programs. And in this observable complex superposition of all kinds of sources, we are now looking for signatures of biological interactions. You see the same cells again. They are from diffuse large B-cell lymphoma. Alone in Germany, there are about 5,000 new cases each year, unfortunately, and if untreated, this disease ends deadly. Already in 2000, Alizadeh and others discovered this cell of origin signature, and they compared it with the gene expression profile of healthy ABC and GCB cells. And once you found such a signature, you can sort new patients, and every column is again the tumor sample of one patient, by that signature, from ABC-like to GCB-like DLBCL, as done here by Visco in 2011. And this signature is clinically highly relevant because despite the identical therapy, ABC-like DLBCL patients only have a 5-year survival of around 40%, whereas on the right side, the survival is around 70%. So we can obviously learn from heterogeneities in the transcriptome datasets. And ultimately, we hope that we can use these heterogeneities to develop more specific therapies, because these patients clearly have distinct diseases on a molecular level. But recall, we first have to find such signatures in a huge and also very complex superposed signal. What do I mean with signal complexity by superposition? Well, let's have a look at the simulation, starting with the zero signal matrix. First, I simulate a signature that looks similar to the cell of origin signature I just showed you for a subset of simulated genes and a subset of simulated samples. And then I destroy information. First, I destroy information of neighboring genes by permuting them. And then I also destroy information of neighboring samples by permuting the columns. Now I superpose a second signature of the same form. And again destroy by permuting gene rows and sample columns the information of neighborhood. And I do this again and again and again, superposing signature by signature, and at the end I also add strong measurement noise. And if I'd shown you this matrix in the beginning, you would probably have said, I can't see any useful structure in there. But, and that's the key message here of the complexity, if you knew at what subset of genes and at what subset of samples to look, in what order of genes and in order of samples to look, then you could actually recover all the simulated 7 signatures. So this matrix contains a lot of information, and that's just 0.2% of the whole transcriptome dataset. In order to detect however, these signatures, we need a search strategy. And we do not only want to discover them, we also want to extract the pure contributions of them to the signal, for which we need a signature model and a regression technique. Okay, now that we know the problem we need to solve and the underlying signal complexity, let's come to the part where I explain you how SDCM actually works. Of course, I can only give you an overview in the next few minutes. If you want to know all the details, have a look at our paper. There you will find a complete mathematical definition of SDCM and quantum mechanics notation, and you will also find a lot of illustrations and comparisons in addition to what I can show you now in the supporting information. Formally, the input signal is a matrix with m gene rows and n sample columns. And we have initial, not very informative, measurement orders I0 of genes and J0 of samples. And I model the signal M as the sum of matrices Ek of the same size plus a noise term eta. And our task is to reverse that sum operator. So to find the summons Ek and also determine the number k hat of summons in the signal that make up the signal M. And of course, that's a highly ambiguous problem, so we need additional mathematical constraints that are sufficient for that dissection. And these constraints should be as flexible as possible in order to fit an as broad as possible range of natural phenomena. Which brings me to my bimonotonic consistency model for the signatures. I demand that for every k, for every signature k, there exists a permutation Ik of gene rows and a permutation jk of sample columns, such that in that permutation, every row of Ek only has monotonic, either increasing or decreasing gene expression values, and also every sample column of Ek has only monotonic values. So now we have defined and know what type of signature and consistency we are looking for. But the mathematically difficult part here are the exist operators, because we first need to find them. Let me now illustrate the 4 key steps of the SDCM algorithm based on this 3D concept signal. The colors are just for you. SDCM only sees the coordinates of the points. First step is to look for an initial representative. Then to optimize the associated gene axis, over which then bimonotonic regression is applied, and then I can dissect the explained signal parts. Let's have a more detailed look on the same 4 steps again. First, I'm looking for an initial representative with help of our signature functional, which gives me a number for any direction A in gene space. And this number is the higher, the more samples exist in this direction and the more correlated they are to the direction. Correlation is measured by x in A over norm x norm A, which you also might know as cosine similarity. But I need a bit more flexibility, which come in form of these dimension weights. All weights together I call the signature focus, and the signature focus and the final converged gene axis are now determined by locally maximizing the signature functional by taking additional representatives into account. Once I have this converged gene axis, I can project all sample points onto it to give me a direction-specific order of samples. Now going to the heat map view. We only have 3 rows in the heat map here because it's a 3-dimensional gene space, x, y, and z, and every column is a point. On the left you see those points with low blue x and y coordinates and high red z coordinates. These correspond to the upper left green dots in the 3D view. Over this signature order, I now apply bimonotonic regression within the signature focus, which results in this bimonotonic matrix where the gray columns correspond to those sample points outside of the signature focus. They do not influence the regression. And now going back from heat map view to 3D view, we can draw this heat map as a curve which looks like this. And at this point, I can again simply subtract the discovered signature and dissect the explained signal parts. And now it's just iteration. We do the same for the red effect and for the offset effect. And then in step 1, SDCM recognizes that there are no more significant signal strengths and no more significant correlations left, and then SDCM terminates. Here you see all 4 discovered signature curves for the concept example again. And let's do a first conceptual comparison. We did in the paper a lot of versatility tests and high-dimensional comparisons with methods like principal components analysis, independent component analysis, non-negative matrix factorization, hierarchical clustering, and a lot of bi-clustering methods like Fabia and Fabia-S. And from all these compared methods, PCA showed the second best sensitivity and specificity after SDCM. So let's compare conceptually with PCA. Of course, you know principal components analysis. It's applied in many fields and quite an old and established method. And for this 3D point cloud, the 3 principal components look like this. The first shows in the direction of maximum variance in the empty space between the simulated red, blue, and green effects. SDCM instead looks for directions of maximum correlation, maximum signature functional. SDCM curves are monotonically nonlinear, whereas principal components are always lines. And principal components analysis uses full projections as a reduction step, which causes all principal components to be orthogonal to each other. So there cannot be more than 3 principal components for any 3D subspace. Also, you have 4 effects simulated in this subspace. SDCM keeps the full space dimensionality for all EK and only dissects the explained signal parts within the discovered signature focus. To test the versatility of SDCM in higher dimensional signals, I've simulated 7 signatures of various shape and form and then randomly superposed them as I showed you in the introduction. This plus added normal noise was then added into SDCM with the task to recover the simulated signatures, which it did, plus a residual signal which was essentially Gaussian noise. And to quantify detection quality, I use such correlation matrices, and the red diagonals mean a high detection sensitivity, and the black off diagonals mean a high detection specificity. So no detected signature axis tries to explain more than one simulated signature. For the same type of signal, a representative result for PCA looks like this. It's also very good in terms of detecting the large signature 1, but it often mixes signatures 2 and 4 and 5 and 6. So sensitivity and specificity of SDCM is higher. Now let's turn up difficulty by simulating 16 times a signature of pattern 3. While SDCM keeps a high sensitivity and specificity, it broke down for PCA, which is likely due to the fact that all signatures now have similar size and cannot easily be separated in terms of variance. And the principal components now likely point again into the empty space between the signatures where the variance is maximal, as I showed you in the 3D concept example. A few more words on bi-dissection is more than clustering or coordinate changes of your point cloud of sample vectors. Here you see the result by hierarchical clustering for the same versatility test signal, and you can see that the dendrograms essentially partition genes and samples by the simulated signature 1, which represented a large overlapping technical bias like labeling protocols. All other similarities simulated by the other signatures are split by these partitions of the overshining signature 1. And that's a problem many cluster methods share because they treat sample vectors as immutable objects and put them as a whole in some similarity function. So they never model or change the values of the components in your sample vectors and then test again for similarities after the change. Another perspective on the same problem based on UMaps. UMaps are very useful to project your high-dimensional point cloud of sample vectors in, for example, 2 dimensions for visualization, and they try to keep the topology. But what is the topology in this 1000-dimensional gene space? Well, you have 2 clearly defined clusters by signature 1, 1/3 of samples and 2/3 of samples. And UMAPs neither have the ability or concept to shift these clusters over another in order to search for similar differences within the two clusters separated by signature 1. So why do we need signal dissection? Well, the idea is to peel off already detected consistent signal parts and then detect and merge initially split and outshine similarities across already detected differences. And the idea or the hope is that this will lead to a higher specificity and correspondence between detected signatures and causal source signals, which in turn will increase the interpretability of resulting signatures, which is key for all biological downstream analysis. I hope I could give you at least an overview of how SDCM works and what special concepts it has. And in the last part of my mini lecture, we'll now have a look on signatures discovered by SDCM for real gene expression data. For all human transcriptome datasets I've dissected so far, SDCM has discovered this strong binary signature. You can identify it by associating with male and female patient annotations as the gender signature. It's useful for control purposes, and it's a first proof of principle that SDCM can detect signatures of biological origin in an unsupervised manner. For DLBCL gene expression, SDCM rediscovered this cell of origin signature known from the introduction. And let's have another look on what monotonicity means. If we slice the matrix at the bottom, then you can see that this single gene is quite noisy over the monotonic trend discovered by SDCM. But the same monotonic trend is shared by all the genes at the same site of the signature. So SDCM works essentially on gene sets level, and that's much more robust than working on single gene level. Another remark, in order to dissect signatures at all, there has to be some distance in the data. So let's draw now this gene from the gender signature over the sample order given by the cell of origin signature. And then you can see no monotonicity is left, no correlation is left, and that's the distance between the signatures. For the same DLBCL data, SDCM also rediscovered the stromal 1 and stromal 2 signatures known from 2008. But as pointed out in the introduction, we are mainly interested in signatures that can predict patient survival. So what I did next is I constructed Cox proportional hazard models for fitting patient survival. And I did so not only for signatures from SDCM, but also for principal components, independent components, and so on. And for any pair of two signatures from any of the methods, I fitted a Cox proportional hazard model. And the best model were these two SDCM signatures by a distance. The second best pair of signatures only had a relative likelihood of explaining patient survival of 0.02. So patient survival is an independent data that was not input into any of the methods, so these two signatures are already validated in the detection cohort as survival signatures. You see the two discovered survival signatures again in the detection cohort. Next, we validated them in two independent DLBCL cohorts, one measured by microarrays, another one measured by RNA sequencing, and you can see the same differences in gene expression there. So now we have two values per patient column from the two signatures, which we can put into our Cox proportional hazard model in order to predict patient survival. And quite strong patient survival differences are predicted and validated in the two validation cohorts, especially if you compare it with the known differences between ABC-like and GCB-like DLBCL. What surprised us most was that when we zoomed into the better surviving subtype of GCB-like DLBCL with an average survival of about 70% after 5 years, if you recall the introduction, then about 1/4 of patients showed significantly inferior survival. These patients are the columns on the right side in both of the discovered signatures for the predictor. And of course, these patients could profit most from a more specific therapy. So we now can use the discovered signature and their top genes and bottom genes in order to give hints for future biological research. On this last slide, let me zoom out a bit. A student team of mine dissected acute myeloid leukemia data in the Beat ML Dream Challenge this spring and made it up here with respect to survival prediction. You have to know that also information about somatic mutations and other things were given in the challenge. We only used the gene expression data for time reasons mainly, and for this the result is quite good. You should keep in mind that SDCM is a mathematically general method. You can dissect any genes cross samples matrix into a sum of overlapping bimonotonic signatures. Just let me give you one example outside of biology. Here we dissected infrared absorption spectra of gas molecules, and the example shown sorts all gas molecules from those having a particular chemical substructure to those without that substructure. And like patient survival earlier, this information is independent, so the signature is validated as of chemical origin. In the future, I would like to dissect heterogeneity across different types of cancer, and we are working to integrate SDCM with biological networks, and of course I'm open to inspiration. If you would like to dissect your own data sets, you can find the complete SDCM source code and toolbox on GitHub. And I'd love if you'd like to follow me on Twitter. This will also be the place where you will get information about new major SDCM versions first, including maybe a Python library next year. Let me close by thanking my current professor and my PhD advisor, Georg and Peter, my friend and good colleague Miroslav, our current PhD students Caroline and Benjamin, with whom I'm working to integrate SDCM with biological networks, my physics master students Joshua, Jonathan, Simon and Karl. And last but not least, let me thank you for your attention. I hope you learned something. I hope to see you immediately afterwards in the FAQ session. And if you have any questions later, feel free to contact me via GitHub, Twitter, or just by email. Thank you very much.